Hello, everyone, and warmly welcome to the first round of the NMR webinar series that we organize within the Treesearch research infrastructure. My name is Anastasia Ryazanova, and I'm a coordinator of the Treesearch research infrastructure. And this webinar series we organize together with Professor Gunnar Westman from Chalmers University of Technology. Today, we have a great pleasure to welcome Diana Bernin, who is Associate Professor at Chalmers University of Technology, who will give us a presentation on the probing porous media and the dynamic processes by diffusion and NMR imaging. Please, Diana, the floor is yours.
Thank you. Thank you so much for starting off this series. So let's see now if I can change the slide. Yes, here you go. So I guess most of you, when you hear the word MRI, you think about images, MRI that is used in the hospital. And often MRI is used in a hospital to look at the brain. And this could be to, for example, look for diseases or to see if the blood flow is working as it should. And so here you have an example of a brain, or basically of a head. And when you look at it, you can see that there are bright regions, but there are also dark regions. And the bright regions, that's the regions where you have a lot of fluid. And that's already an important part about the MRI. We measure only things that are mobile or that are liquid. So in this case here, you will image the water and you don't see the bones because the bones are solid. So MRI, there are more developments ongoing, but it's basically measures what is liquid. Then I would like to show you another image, which you might see what it is. It's an egg, which has been cooked, but not cooked until it's completely hard. So it's still soft in some parts. So you can see here the edge, the dark blue color. That's where the protein is hard. But then you have still a lot of water in the protein, so you get some kind of signal. And then here you have a gradient of different colors. And this is the egg white. And if you normally open an egg, so you can see that there are different, some egg white is more liquid and some egg white is more solid. And then here you have the egg yolk. And sometimes when you eat a cooked egg, you can also see that there is something in the middle of the egg yolk, which you also can see here. So the important feature or the important things that I want you to notice here is that here we have a signal intensity. So, and that's really important to understand when you have an image, is that if we want to see some things, we have to have a contrast. Otherwise, the image would be completely blue. And I will tell you in a couple of minutes what gives you a contrast in an image, and in particular in an MRI image. This is maybe more familiar to you. That's an MRI image of a piece of wood. The wood is immersed in water, and it's in a cup. So here outside this red ring, it's water, but then you have here the wood, which has been soaked in water for a while. And already here you can see different features. You can see here these different rings. And you have like, here is something red, and here it's something more dark. And another important, when we zoom in on this image, then you can see that these are squares. So these squares we call in the MRI language like voxels, which is basically a volume element of a pixel. So a pixel is 2D, but a voxel, since in MRI you always have like a slice thickness, it's a volume element. And these gives you also the resolution of an image. So when you image, for example, you image a sample which is 1 centimeter, and you say you want to have 10 pixels, voxels, then you have a resolution of 100 microns. And if you have 100, then you have like 10 microns. So this is very important to keep in mind. Then I always like to think about, when I do MRI or NMR measurements, about that I have spice in my samples. And this spice is the proton, which has a nuclear spin. And here you have a water molecule, and you have 2 protons on this ones, or 2 hydrogens, if you would say so. And why do I call it spice? Yeah, if you have water in a glass, you can see through it. That's quite easy. But most of the samples that we would like to study, we do not see through it. Although MRI and NMR can measure and see what is inside your sample. So that's why I call it spice. Also think about the brain. We can see into the brain. We can get an image of the brain without opening it up. So the person is still alive after an MRI scan. But again, what gives you this contrast? So here I indicated 2 different voxels. And in this voxel, I have 3 water molecules. And in this voxel, I have one water molecule. So if I would now run an MRI on this one, I would get a dark spot here and a brighter spot here. And why is this? Because it's the amount of hydrogens or protons that I have in this voxel. So here I have 6, and here I have 2. So 2 spice and 6 spice. And that means that the difference, that gives the contrast in an image. And here the difference could be the amount, the density, how many I have. But then what makes a difference? For those that are used or have been running NMR before, we know that we also can run diffusion with NMR. And we can do the same also with MRI. And here I have indicated a water molecule, which is confined in another square. So it cannot move out of this square. So here the motion or the, the transport, the diffusion is hindered or maybe even completely restricted. So another contrast in an MRI image might be diffusion, the mass transport. So then it wouldn't be the amount, but it would be how fast or how far molecules can travel. And this would be called diffusion-weighted imaging, for example. Then we have another important parameter, which I call chemical interaction. In NMR language, you would call it relaxation times. You have different ones, the T1 and T2. And by this, I mean, if this water molecule, for example, likes to interact with, let's say, an interface, or with polymers that have hydroxyl groups on, then these relaxation times will be altered, and then you can use these to make a contrast and to make a difference in an image. And this will be called then relaxation-weighted imaging. Then there is even another dimension of difference. So getting back to the spectroscopy, the NMR again, you can also get information about what's inside a voxel, not only about the amount, but also what is inside. For example, here you have a water molecule and you have another molecule. And you can require a spectra in these voxels, which would then look like this. So here you see, you get all information you need. That's only water, but in this voxel, we actually have more than water. Yes, so that was basically about the theory that I would like to tell you. And because I don't want to bore you with a lot of equations and stuff. But if you want to know more about the theory, you can write me an email and then I can give you some sources, or we can also discuss more about it. So today I would rather like to inspire you with the applications that have been performed in my lab. And this does not mean that there are other applications. MRI can be applied to many, many, many things. But I will tell you also a lot of preliminary results and what can be challenging. So I will talk about packaging materials, 3D printed cellulose materials, the cellulosic materials, about timber and about filtration, but also about cellulose coagulation, which are used to make, for example, textile fibers, if you think about the man-made ones. And then for sure, everybody talks about lignin, but I will only mention briefly how one could think about lignin. And I think there will be excellent talks about lignin later in this series. So paper packaging, for those that do not work in this industry, so there are many, many parameters that impacts a paper, how it behaves and how it takes up water. Now we have to replace all the paper packaging with all the plastic with paper. And for this, we have to make a rather hydrophilic product into a hydrophobic one. And this you do by, for example, adding chemicals, which is called sizing. But then how water is transported or any other liquid into this paper packaging material, it might also depend on the production process of the pulp, but then also on the production techniques. For example, if it's wet or dry formed. And here you have an example of a dry formed packaging process, which I would like to point out has 75% less energy consumption. Then when you think about the paper straw, it's layers and it's glued together, and this glue is most likely a hydrophilic glue. And it's not very nice if you would like to drink a cocktail and the paper straw is dissolving after 5 minutes and you have to hurry up. And then also the bleaching has an impact. And here you also have an indication of if it's a machine direction and so on. And here you have an example of the difference between an unbleached and a bleached pulp surface. So when you work in the paper industry, you have most likely seen this COP test, which is illustrated here. It's 60 seconds normally, and then it gives you a value. And so you quantify, or you know if a paper is more or less hydrophobic depending on this value. Then we have been contacted by Tetra Pak, and we initiated a master thesis. And this has been very successful. And now we are running a lot of different samples. And so Tetra Pak is mainly interested, or at least what I know, in the transport through the edges, but also through the plane here. So we tried to mimic this COP test with MRI and see how water is transporting into this paper material. And it's a very thin one. So then we started off by thinking, okay, since it's a very thin paper, so we will have very little water, and they're also hydrophobic because their size, these materials, we would like to have a large area as possible, because then we will get as much signal as possible. And in our MRI that we have, which is dedicated for chemical engineering applications, the maximum size that we can measure is 66 millimeter in diameter, so which is quite large. NMR tubes, 5 millimeter, but here it's 66 millimeters. But then already the first problem started. Paper wants to buckle. And if it buckles, it gives a problem for us to know where the paper is, or if it's, so it's not easy to interpret the data we get. So the students came up, the really smart students came up with another idea. So here you can see our first approach. We have this holder in which we add the paper. But instead of having water added on top of this one, we add a rubber, and the rubber has a square in the middle. And in this square, we add another 3D printed reservoir holder. And it's really good with 3D printing for the MRI, because it helps us to adjust our rigs that we have to do all the time very easily, and it's very fast. So it's very good for us. And this water reservoir is also important, because we don't want to get water that is getting from the sides. We really want to study the water transport from this square into the paper. And then we have a lid, and this lid is going to exert some pressure both on this holder and also on the rubber, to make sure that water is not going anywhere else where it should go. And here this lid has the option also to add water inside the magnet, so that you don't need to, so that we can basically measure it in situ. Here you have an MRI image of this square here. These lines are used to only for orientation. And in the end, to measure fast, MRI is a rather slow technique. And images take a long time, especially when to get them with a good resolution. And you remember that the paper size or thickness is only like 400 or 500 microns. So we would like to have as many points as possible in the paper. And for this, we need to measure rather fast. Then we average over this area. So we take a profile, we basically collapse this dimension. So we only have the average in this. Oh, sorry. And then it looks like this. So it's called 1H profiling. It's still like an imaging technique, but it's optimized to be fast and also to get a rather good resolution, which is here 31 micron. And here you have, you start from the bottom, you have no signal, and then here the paper is, and then here you see the water which is in this square. And then here you have the time in this dimension. So we repeat the same experiment over and over and over again. And what do we see? Then we start to see that water is ingressing into the paper material, because here we can see a signal build up. So this is when water progresses into the paper. But then, as I said before, that its paper is prone to buckle and some packaging material also to swell. We have another motion, a movement, which is that the waterfront is pushed upwards. And that's because the material is swelling. So we have one movement that direction and the other one in this direction. And that's what we will compare now for different packaging materials. Here you have an example how it has been swelling after the reaction is finished or the experiments are finished. So here it's again the swelling, which is the motion, the movement that goes and that pushes the waterfront in that direction. And here we have what the water goes inside. And here you see the distance that the front has been moving with time. So here we compare the wet formed with a dry formed packaging material. And you can already see that this one has only been swelling like 250 microns, whereas here it has been swelling 600 microns. But then we need to figure out how to normalize this because the initial thickness is also different. So that's something that we have to look at. Another very important difference is that here the water goes in very, very fast. Already after 25 minutes, the saturation is basically reached. Whereas here, for the dry formed packaging material, it takes much longer time for the water to be completely saturated in the material. Then about sizing, I got two different, or I got a couple of packages, dry formed, with different sizing. I don't know exactly how much because that's an industrial product. But here it's obvious that it seems that maybe more is not better, because here even the less already shows exactly the same behavior as it does with the more. But this doesn't mean that it works also for oil and other liquids. So that would be interesting to test next is, instead of water, to look at oil and emulsions, for example, some kind of dressing when you think about salad, or also like coffee and other things. But this technique seems to be rather good in picking up this dynamics, how the liquid is transported into these materials. Then I also mentioned that it is very interesting to see if there is an impact on the production process of these packaging materials. So we received a couple of trays, as you can see here. And then we took samples from the bottom, but we also took samples from the sides. And there is a big difference. So most likely the mechanical interaction, it's very important to look at if to understand what the reason for this is. So when we look at the bottom, we can see that the swelling is huge in comparison to the side. And also the swelling is much, much faster in comparison to the side. Then the water behavior is rather similar, which is a good thing. But one would still like to understand why it swells more at the bottom in comparison to the side. Maybe it's because the fibers get orientated when you press it into this form, or that the different pressures give a different porosity in these different regions. We don't know, but that's one of the questions we are looking into currently. Yeah, and then we have the paperboard. So I'm not sure if you're aware of this, but most paperboards are often like layered materials. So here you have a brown layer and you have a white layer, and often they're glued together with some glue that is most likely starch-based. Then you have also, this one is for sure bleached, and the other one is it's not bleached or less bleached. And then often on the white side, you even have some kind of calcium carbonate layer to be able to print on it. And when we compare the glossy side with the brown side, we can see that it swells much more for the glossy side in comparison to the brown side. And the water travels far less, or here when you have the brown side, it travels much further into the paperboard in comparison when you start from the white side, which is kind of interesting. So somehow there seems to be a barrier when you start from one side comparison to the other side. So now let's switch gears and look at other applications. So here we have a 3D printing robot that can be used to print building structures. And we are working together with the Chalmers, like the architecture and the building divisions. So you have these pellets which are made of cellulose and plastic, and then it's extruded to this one, and then you print this layer by layer by layer. This material has been used, it's for in-house use, but it would still be very interesting to understand if water is going to be, how it behaves when it's exposed to water. And for this, MRI is again a very good technique. So we soak, now this time we had not to build any MRI rig because we could only immerse one of these pieces, not this big one, but a part of it into the water and then see how it behaves. So here it has been soaked, and it's quite obvious that we do not really see that the water is going to be in the, where it's more dense. It's basically only swelling on the sides. And since there is cellulose inside, you can actually see that these cellulose fibers are, they really like stick out. So I assume that most of this, most of water that has been taken up here is from the cellulose, which is exposed. And then you can see that it dries with time, as expected. So we will now look into this further by making different pellets with different materials, because this has been pellets which are made by Stora Enso, so we don't know exactly what's inside and which ratio it is. But we would like to look further into this and see how it behaves. If it's only on the surface, which is here, then most likely you could only paint it, and then you might have a quite good barrier against the water. Then in the same collaboration we have, there is another research question. And how, when you have timber, which you have, and you add concrete on top, how water, if the water is going to be taken up by the timber, then there will be less water for the concrete to harden. And it will also impact, because the wood will swell, maybe some mechanical strength. So here we had to build a rig again, how to keep this in place, make sure that water is not moving where it shouldn't be, and also how to add the concrete. So here is the picture that you have already, that you saw before in the first slide. And here we are closest to the water, but then here we go further into the wood, or into the timber. And it's quite interesting that it seems that here the water is not equally distributed. Here we have a lot of water, but here are regions where it's much less water. And it would be very interesting to understand why in some regions there is more water uptake, where in others it's not. And here you can also see air bubbles when you look at it. And then, so this is a start. This project has started, so we don't have so many results on it yet, and it's not so easy to measure on concrete as such. But here is another way of looking at it, not like the axial slides that you have seen before. These ones are sagittal ones. That's how you call it. That's hospital language, but that's also like the chemical engineering MRI saying coronal and sagittal ones. So here we start from the edge, then we go into the middle, and then we again to the edge. So you can also make like 3D images, but MRI takes time. So if you have something which happens fast, then it's not a good idea to do 3D images. But here you can clearly see that the water here has traveled much further than here. And also here you have some kind of channels where the water has been traveling, but not here, but only in these channels. And when you look at this edge in comparison to this edge, it seems there's much more water here. Yeah, then also a started project and together with Hans Teljander, but he's soon to be retired, and I hope we can pick up on this. It's about filtration. And also here, we had to build a rig first. So this is a long tube. It has a top where we can put the pressure on it. And then here at the bottom, where we actually measure, we can add filter paper or membranes or different things. So this we have put into the MRI. It's actually already, it takes some effort to get this done into the MRI. So it's good you don't do this every day, but it's possible. And here is one example. This was a static one, so it was after we have been filtering. And here the filter cakes looks really good. So we have the filter cake here. It's darker because it has less water. And here we have the water on top. Looking about what could be giving contrast, I made some tests, and it's obviously that the relaxation time, the T2, is a very good way to contrast what is happening in the filter cake, but also the diffusion. The diffusion is much lower in the cake than it is where the water is. And then here we have done it during filtration. And I know these pictures are not very good, so that's also why we need to redo it. But you can definitely see there's a lot of structure happening when we do the filtration. So I think it is a very good technique, and we hopefully can also measure flow when we do the filtration. But this it's always when you start a project MRI, you need to know a lot of things about your system before. Which time scale things are happening, and then you have to design the MRI experiments according to this. There are so many pulse sequences available, the same also with NMR, so it's very good to know what you're looking for before you start. Yeah, and then another project, which we look into the mass transport during coagulation. And we cannot measure on the fibers during the fiber production, because the fibers are too tiny. And the resolution is often the best one you can get, maybe is 10 microns. So what we instead did, we also built another rig. So we used the sausage model. I don't know if you have heard, people at RISE call it also sausage model. And it's you add you have to dissolve the cellulose and also the lignin. Here it's aiming at to make bio-based carbon fibers. So you add the cellulose lignin dissolution solution here. You get a film on this piston. You add this piston, and then you add water or any other solvent, which is an anti-solvent that makes the cellulose to coagulate. And we believe that we believe that the mass transport, because the solvent has to go out and the anti-solvent has to go in, might impact the crystallinity of cellulose. Also, if there will be inclusions, because depending on how fast things are, and so on. So we try to measure this, but it's a kind of it's a multi-phase system. So imagine you have two liquids which are both transparent mixing. And to show you how it looks like, here we have a movie. It works. So you can see that MEMAC, which is the solvent here, is coming out, but also parts of lignin. And then we have been struggling quite for some while to figure out how can we measure this? How can we use MRI to figure out if it's different when you use different solvents and when is it finished? So here we use the technique which is called saturation slices. So in the beginning of the coagulation, you can see that, so when there's no mixing, then there are basically only stripes, and the stripes are straight. But if you have mixing, then these stripes will show the pattern, what is happening. So for Jan, my postdoc, she did that for coagulation in water, for cellulose, but also for having different low molecular weight fraction lignin and high molecular weight fractions, and then also for in ethanol. And you can see that these, and we have multiple images. It's again the question how to analyze those. But you can see that there's a lot more mixing ongoing when you have the coagulation happening in ethanol in comparison when you do it in water. And so she tried to treat these images by when it's black or white, that you put like a 1 and a 0, and then see when the slices get stable. And then from this, estimate kind of a time frame when there is no longer any mass transport happening, so when everything is in equilibrium. But you can also do, I don't know if you remember, but in the beginning I told you can even do like spectroscopic voxel imaging. So another way to see if the mixing is finished is to see how the concentration of MEMAC, the solvent, is changing with time. So here she recorded images in this little voxel, not images, spectra, and she did that with time. And then here you can see that MEMAC, so these are the, if you know NMR, so these are the peaks from the MEMAC, and they're increasing time. And then you can integrate this peak, and you can plot this with time, and you will get a very similar time frame. It agrees very well to what we see when the slices stabilize, so when the lines are straight. Then I continue with the spectroscopic part here, because there is also some imaging, which you might call NMR imaging, that you can use when you have normal probes. You must have gradients on it, but you can do it in NMR tubes, and you don't need to have a dedicated MRI machine that we have at Chalmers. This was my first paper, and I figured out that I don't have the original images any longer. Most likely they got lost somewhere, so I had to copy them from the paper as such. So if the quality is not very good, I'm sorry. But it's again about coagulation of cellulose. But this time we have used a different solvent. This solvent contains fluoride ions. And fluoride is another nuclei, which is very good to run NMR on. And there are a couple more. You can even do fluoride imaging. You can also do sodium imaging. You can do, there are some phosphorus imaging, for example, would also work. There are some nuclei which work very well. But here we have a cellulose solution. And then we add water on top. Again, and it's the same. It also coagulates cellulose. So and then this, you record these 2D sequences, use them, and you basically get spectra that are stuck. So it's each of this is a spectra, but you can plot this in this 2D mode and it's the intensity that you see here. So then you have water here, and here you have the cellulose peaks, and then you can do the same for the fluorine. This was 2 different experiments. In this probe, we could not. I don't think we have recorded it in the same. So we have had one run measuring the fluoride and one run measuring the proton. But if you have, there are probes, or consoles, I would say, hardware, where you can measure fluoride and proton at the same time. So then you would measure like proton and then fluoride and proton and fluoride without needing to change the sample. And the fluoride ions are very sensitive. So you can see when it's the water, it has a completely different chemical shift compared to when it is in this solvent. And then you can see the water, it's very broad because it's interacting also a lot, moving further and further and further. And that's another way not to measure the diffusion, but you can extract from this how fast the front is moving. And we did that here too, both for the fluoride ions and for the water. So if you want to know more, you can have a look at this. Yeah, and as I promised, lignin. So diffusion can be very useful to run on lignin too. We have been able, and you can find this here in the paper, we are not first, but to measure the diffusion of lignin and then convert this to a molecular size distribution. And then Elodie and Bruno, they made also some GPC, and they got very similar values. So it was kind of very nice. So if in case you wouldn't have this instrument, you could still do this one, but it's kind of tedious to do all the analysis and to make sure that you have to measure very accurately to make sure that this is really reflecting the right size or the molecular weight distribution. And it's also, so you have to convert it. You always have to normalize it to something, or you have to compare it to some other molecules. And there is always a risk of that this molecule is not reflecting the same behavior as lignin does. And yeah, and it might also be tricky to see if there would be some small bumps or so, but in principle, it's possible. Then last slide here. Excuse me. I mean, we talked a lot about diffusion too, and also the talk should be about diffusion, but it's, however, very, very difficult to measure diffusion inside paper or wood-based material as such. And that's because the water is so, the relaxation times are very, very short, and that there is a lot of problems, I wouldn't call it problems, but that you have to take care of, that the water is interacting with the cellulose, for example, through the hydroxyl groups. And this will also add on as another contribution that might alter your diffusion coefficient. So if you want to find how to do it in a good way, then you should look at this paper, because there is everything explained in detail, and so it's a really good one. But it's quite tedious too, because you have to record a lot of different sequences and then basically subtract the interactions of the water with the cellulose from the diffusion. Yeah, and with this, I would like to acknowledge both the funding we got, and then also many in my group, but also Tetra Pak and Yangi. And with this, I would like to thank you for your attention.